Welcome, everyone. I'm Greg Slobodkin, editor-in-chief of scienceboard.net. And as part of our continuing coverage of ISCT 2022 in San Francisco, we are honored to have with us Bruce Levine, who is president of ISCT. Welcome, Bruce.
Hey, Greg. It's great to be with you virtually.
Virtually, yes, indeed. It's great to see the conference celebrating a major milestone. I mean, not only the fact that folks are actually gathering, but also it's the 30th anniversary of the founding of ISCT. This is an interesting, time to sort of reflect on the state of the industry and, get an assessment of where we're at. And so what can you say to us in terms of the overall status of the field as you see it? Where are we at and where do we need to go?
Yeah, Greg, 30 years ago, ISCT was started by a small group of individuals that were manipulating blood stem cells for stem cell transplants, and it has evolved into a multifaceted organization involved in all types of cell and gene therapy. And we are now built on three interdigitating pillars. We have scientific committees, we have regulatory and quality operations, and we have commercialization. So just as the field has matured and we have multiple approved products that are available for patients, ISCT has matured as an organization, and we've grown by leaps and bounds, especially over the past few years. Now, if I go back to those three pillars that I mentioned, I've likened those to the Rosetta Stone. So just as you needed Greek to decipher hieroglyphs, you need to be proficient in regulatory and quality operations to move the science to commercialization. For commercialization, you need a sound backing in science. So everything has to work together. And the beauty of ISCT is we're all here speaking to one another and learning from one another and learning from all of the regions.
Very good. I mean, the other thing I noticed, the theme of this year's conference is, bench to bedside to benefit. And so looking at that, what do you think it'll take to see a wider array of cell and gene therapies available to address multiple diseases in larger patient populations?
Yeah, Greg, let me take the format of the meeting. What we did this year was rather than specific cell type or disease area, we elected in an experiment to follow the translational pathway. So from innovation to translation, first in human, expanding that, and then wider patient access. And all of the cell types and disease areas are represented within there. We feel it's a logical flow and allows people with different expertise to learn from one another. Now, to your question, what will it take for more? I think we're making progress, especially over the past few years with the regulatory approvals and CAR T cells. I think we'll have some approvals in a T cell receptor therapy and maybe tumor infiltrating lymphocytes, and we will have gene editing blood stem cells for the hemoglobinopathies, and we'll progress from there. So there is progress, and what it will take to accelerate that, I think most people that I speak to at or near the top of their list is workforce education and training. There's a talent shortage because the field has grown so quickly. There just aren't enough people with expertise in cell and gene therapy to fill all of the roles. And this starts at the bench, at the entry-level jobs, and it goes all the way up to the C-suite. And that's really where we can play a huge part, ISCT, in our educational programs and bringing more people in to the field.
It's interesting. With ISCT celebrating 30 years since its founding, it also gives you some perspective. We're talking about cell and gene therapy as an industry is kind of in the nascent phase, is it not? Compared to other modalities. And so, as you learn more and the number of therapeutics receive regulatory approval, do you expect the targeted indications to increase? And if so, where and what kind of patient populations would you would you envision those encompassing?
Yeah, I think those are going to increase. I mean, just yesterday, Novartis received approval in Europe for follicular lymphoma. We have some very promising data in solid tumors. I mentioned the data in the hemoglobinopathies. I think those are going to be moving along, as well as other indications. But to the essence of your question is, what do we need to get in place for the arrival of these therapies? You have to think of the infrastructure, and that starts with the collection of the cells and qualification of leukapheresis centers, the logistics to ship the cells to manufacturing and the testing and receipt of the cells, and educating clinicians and pharmacists. So it's a whole gamut of clinical, logistical, manufacturing expertise. And I liken it to think about, say, the year 1900, 1905, 1910, the automobile industry. How many gas stations were there? How many automobile repair shops were there? How many programs were there for educating automobile technicians? So that's the stage that we're in now. We're making discoveries. We have regulatory approvals. We have patient benefit. And this is just going to expand and expand and expand. And we're building that infrastructure with the goal of benefiting patients worldwide.
Yeah, I mean, it's interesting. I mean, one of the challenges for CGT has been in the area of manufacturing. And I saw an announcement recently from Penn, where you're a professor of cancer gene therapy. They have actually shortened significantly the manufacturing time for CAR T cells. I mean, down to, I think, 24 hours versus days to even weeks. Talk to me a little bit about the significance of that.
Yeah, sure. So that 24-hour paper, that's just preclinical, just to clarify that. Where our standard process is for the approved products is in the range of 9 or 10 days. A few years ago, we published a paper out of Penn showing that if you reduce that to 5 days or 3 days on a per cell basis, those CAR T cells are more potent. We actually have a clinical trial open now with that 3-day process with a little bit of a twist. The cells also produce a potency-enhancing cytokine IL-18. So we have early data. It's not yet been presented publicly, but we're very excited about it. Now, to your point about the 24-hour, that will take some time to get into clinical trials. It's also a regulatory question, right? So how do you think about doing all the testing for a cell product that at that point is really still changing? How do you look at the integrated or non-integrated virus, the expression of the CAR on the surface, potency? So those are all open questions that we will need to address to get into clinical trials. However, I will say that there is a Novartis process that is a 48-hour, 2-day process, and that will be presented at the meeting in a session on advanced CAR T-cell manufacturing 2.0. We have a talk from Novartis and a talk from Kite on their advanced manufacturing. So this work is going on, and in some cases, patients are already being treated, and I think that's going to be the next wave, is to improve the manufacturing so that we can get these products to patients more quickly and also reduce costs.
Yeah, I mean, the other thing on the manufacturing front that we hear so much as regards to cell and gene therapy is cost. It costs so much to manufacture. Is there room for improvement there, and are you optimistic that costs can be brought down significantly?
Yeah, there's certainly room for improvement. There are a lot of reagents and materials that go into manufacture. Many of those are sole source. We need to bring in more tools and equipment and reagent suppliers, but also shortening the culture and implementing automation assists in reducing the cost. But we could even go one step beyond 24 hours. And there is work on in vivo gene therapy, and you can deliver the the genetic material encoding a CAR in a virus or in a nanoparticle or in a lipid nanoparticle, as we published out of Penn, worked with a targeted lipid nanoparticle, delivering a CAR directed against fibroblast activation protein in a mouse model of cardiac fibrosis. So I think we're going to see some really exciting developments, not only in manufacturing, but in thinking about delivery and in thinking about combinations and enhancing potency, because you have to think about cells as drug factories, and we can program them. And it's just learning how to do that to address the pathology.
Yeah, and the other thing is, I mean, your work at Penn with others have been a source of incredible innovation in this field. Can you talk a little bit about the spinoff that you helped to co-found as a scientific co-founder?
Yeah, we have a number of spinouts from Penn, the Center for Cellular Immunotherapies. I'm co-founder of two. One is Community Therapeutics, which is investigating CAR T cells in solid tumors. And the second is Capstan Therapeutics, and that incorporates the targeted lipid nanoparticle technology that I just described.
Very good. And so your co-founder on that at the Community is who?
So the Community, they're separate companies, right? So Community co-founders is myself, Carl June, Jim Riley, and Chu, and Capstan co-founders are myself, John Epstein, Drew Weissman, who everyone knows from his RNA work, Carl June, as well as Steve Albelda, Ellen Pure, experts in fibroblast activation protein and fibrosis, and Hamida Parhiz. And we have a co-founder from John's lab, Haig Akajanian, who's now at the company.
Very good. And your term as ISCT president is through the end of this year?
Well, we can actually measure it in hours. It will be at the end of this meeting.
Okay. All right. So it's soon upon us. Well,
yes.
I appreciate your time and wish you all the best. And it's amazing to see the growth of ISCT over these years, and we'll be continuing to cover.
Yeah, thank you very much. Look forward to it.